You will die without it. Not eventually, not theoretically, but absolutely and with certainty. Stay awake long enough, and your body will shut down. Your immune system will collapse. Your brain will lose the ability to regulate even basic functions. Within days, you'll develop tremors, hallucinations, and paranoia. Within weeks, you'd be dead. In the entire animal kingdom, from fruit flies to elephants, from fish to birds, no creature has ever been found that doesn't sleep or engage in something remarkably like it. The longest scientifically documented period a human has gone without sleep is 11 days, achieved in 1964 by a 17-year-old named Randy Gardner. By the end, he couldn't complete simple tasks, mistook a street sign for a person, and suffered from severe cognitive impairment. Yet the moment he was allowed to sleep, his brain began its urgent work of recovery. Here's what makes this terrifying. We still don't know exactly why. After more than a century of scientific investigation, thousands of studies, and revolutionary discoveries about the brain, we cannot point to a single, definitive reason why losing consciousness every night is so essential that evolution has made it non-negotiable across every species studied. Sleep makes you vulnerable. You can't eat, reproduce, or watch for predators. Natural selection should have eliminated such a dangerous behavior unless the alternative, staying awake, was even more lethal. The answer, researchers now believe, is that sleep isn't one thing serving one purpose. It's a cascade of vital processes that can only happen when consciousness releases its grip. Your brain, during waking hours, is like a city during rush hour, too busy with immediate demands to do maintenance. Sleep is when the traffic stops, the lights dim, and repair crews emerge to fix roads, clear waste, and prepare infrastructure for tomorrow. Except in this case, the city is your mind, and the repairs are molecular, cellular, electrical, happening at scales from individual synapses to entire brain networks. What you're about to discover will change how you think about those hours you spend in darkness. We'll journey through the hidden architecture of sleep, from its distinct stages to the chemicals that build and release sleep pressure throughout the day. We'll explore the glymphatic system, discovered only recently, that literally rinses your brain of toxic proteins. We'll see how memories are filed away, how emotions are processed, and why dreams might not be the mind wandering, but the mind working. And at the end, we'll circle back to that original question, why do we sleep? And find that the answer might be more profound than anyone imagined. Hello there and welcome to the Sleepy Science channel. I'm so glad you found your way here. Maybe you've had a long day, or perhaps you're simply curious about what happens when you close your eyes each night. Either way, you're in the right place. Tonight's exploration is gentle, thorough, and designed to let your mind drift as we uncover the hidden world of sleep. So find a comfortable position, let your breathing slow, and allow yourself to sink deeper into rest as we begin this journey through the science of something you do every single night. If these calm explorations help you unwind or give you something fascinating to think about as you drift off, consider liking and subscribing to the channel. It helps others find their way here too, one sleepy soul at a time. Now, let's begin. What is sleep? The architecture of rest. Sleep is a naturally recurring state of reduced consciousness and decreased responsiveness to external stimuli. But this simple definition barely scratches the surface of what sleep actually is. To understand sleep, we must first understand that it is not a single uniform state. Rather, sleep is composed of distinct stages, each with unique patterns of brain activity, each serving different functions. When you drift off to sleep, you don't simply switch from wakefulness to unconsciousness like flipping a light switch. Instead, you descend through a carefully orchestrated sequence of stages, cycling through them multiple times throughout the night. According to research published in Stat Pearls, sleep occurs in 5 stages: wake, N1, N2, N3, and REM sleep. Stages N1 through N3 are collectively known as non-rapid eye movement, or NREM sleep, representing progressively deeper levels of rest. Each complete cycle through these stages takes approximately 90 to 120 minutes, and most people experience 4 to 6 complete cycles during a typical night of sleep. The journey into sleep begins with stage N1, the lightest stage of NREM sleep. This is the threshold, the border zone between wakefulness and sleep, lasting only about 5 to 10 minutes. During N1, your brain waves shift from the rapid beta waves of alert wakefulness to slower alpha and theta waves. Your eyes may move slowly beneath closed lids, and your muscles begin to release their tension. You're easily awakened during this stage, and if someone wakes you, you might insist you were never truly asleep at all. Many people experience sudden muscle contractions called hypnic jerks during this stage. That startling sensation of falling that jolts you awake just as you're drifting off, your body's last protest before surrendering to sleep. From N1, you descend into stage N2, which represents true sleep. This stage accounts for approximately 45 to 50% of your total sleep time, making it the most substantial portion of the night. During N2 sleep, your heart rate slows to a steadier rhythm, your body temperature drops by about 1 degree, and your muscles relax further. But what makes N2 sleep particularly fascinating are the distinctive electrical patterns that appear in brainwave recordings. Sleep spindles, brief bursts of rhythmic brain activity lasting 1 or 2 seconds, sweep across the brain like waves crossing a still pond. These spindles, as research from Nature Scientific Reports has shown, play crucial roles in protecting sleep from external disturbances and in consolidating memories, acting as a kind of shield that keeps you asleep even when sounds occur in your environment. Alongside spindles, large, slow waves called K-complexes appear spontaneously or in response to sounds. These K-complexes seem to help the brain suppress responses to external stimuli that might otherwise wake you, like a biological noise-cancelling system. Then comes N3, often called deep sleep or slow wave sleep, and this is where the body descends into its most profound rest. This is the stage where your body does much of its physical restoration. During N3, your brain produces large, slow delta waves, like great rolling swells crossing a calm ocean. These waves are so large they represent millions of neurons firing in perfect synchrony. Your blood pressure drops by 10 to 20 points, breathing becomes deep and regular, and growth hormone is released to repair tissues and build bone and muscle. The arousal threshold is highest during N3, meaning it's remarkably difficult to wake someone from this stage. If awakened, they often experience sleep inertia, a state of confusion and grogginess that can last up to 30 minutes, as if consciousness needs time to fully reassemble itself. In adults, N3 sleep makes up about 25% of total sleep time, but this percentage decreases with age. Young children spend much more time in deep sleep, sometimes 40% or more, while elderly adults may have very little or none at all. The final stage in each cycle is REM sleep, named for the rapid eye movements that occur beneath closed eyelids. REM sleep is paradoxical. While your body lies still, your brain becomes remarkably active, with patterns of brain activity that closely resemble wakefulness. According to research from the National Heart, Lung, and Blood Institute, during REM sleep, your eyes dart rapidly in various directions, your breathing becomes fast and irregular, your heart rate increases, and blood pressure rises. Yet, despite this inner storm of activity, your voluntary muscles experience temporary paralysis, a state called atonia. Only the muscles controlling eye movement and breathing remain active. This paralysis is protective, preventing you from acting out the vivid dreams that occur predominantly during REM sleep. REM periods follow a distinct pattern across the night. Your first REM episode may last only 10 minutes, but as the night progresses, REM stages grow longer, with the final one potentially lasting up to an hour. In total, REM sleep accounts for about 20 to 25% of adult sleep time. Newborns spend as much as 50% of their sleep in REM, a proportion that decreases through childhood and continues to decline slightly with aging. The cycling between NREM and REM sleep follows predictable patterns, but with important variations. Early in the night, you spend more time in deep N3 sleep and less in REM. As dawn approaches, N3 stages shorten or disappear entirely, while REM periods lengthen. This changing architecture across the night reflects different functions being performed at different times. Early night deep sleep appears especially important for physical restoration and certain types of memory consolidation, while late night REM sleep may be crucial for emotional processing and creative problem solving. This architecture, the structure and organization of sleep stages across the night, is not arbitrary. It has been refined over millions of years of evolution, shaped by the needs of the brain and body. When sleep architecture is disrupted, whether by sleep disorders, shift work, or other factors, the consequences ripple through every aspect of health and functioning. The stages of sleep are not simply different depths of unconsciousness. They are distinct states, each with its own purpose, its own patterns of brain activity, and its own contribution to the restorative power of sleep. The brain during sleep. To truly understand sleep, we must look beyond behavior and examine what happens inside the skull during those quiet hours. The sleeping brain is not a dormant brain. It is a brain transformed, operating under different rules, serving different functions, but working no less hard than during wakefulness. One of the most powerful tools for studying the sleeping brain is electroencephalography, or EEG, which measures electrical activity by placing sensors on the scalp. These sensors detect the synchronized firing of millions of neurons, producing wave patterns that characterize each sleep stage. During waking alertness, the EEG shows fast, low-amplitude beta waves, reflecting the rapid and varied processing of sensory information and thoughts. As you relax with eyes closed, these give way to slightly slower alpha waves, a sign of calm but awake attention. When sleep begins and you enter N1, alpha waves diminish and slower theta waves emerge. The brain's activity becomes less varied, more synchronized. In N2 sleep, those distinctive sleep spindles appear, generated by circuits connecting the thalamus and cortex. These spindles represent moments when groups of neurons fire in rapid, coordinated bursts. According to research published in the journal Nature Neuroscience, these spindles may help the brain gate sensory information, selectively allowing some signals through while blocking others, thereby protecting sleep from disruption. But it's in N3 sleep that the EEG reveals its most dramatic transformation. Slow, high-amplitude delta waves dominate the recording, waves so large and slow they represent vast populations of neurons firing together in a synchronized rhythm. These slow oscillations, as research from multiple laboratories has shown, appear to orchestrate the brain's nighttime activities. They coordinate the transfer of memories from temporary to permanent storage. They facilitate the clearance of metabolic waste, and they may allow for synaptic downscaling, a process where overactive neural connections are pruned back to more efficient levels. Then, as the brain transitions into REM sleep, the EEG undergoes another dramatic shift. The slow waves vanish, replaced by fast, low-amplitude activity that looks remarkably similar to the waking state. This is why REM sleep is sometimes called paradoxical sleep. The brain appears awake, yet consciousness is profoundly altered. During REM, specific patterns appear, including sawtooth waves and theta rhythms, particularly strong in the hippocampus, the brain's memory center. But electrical patterns tell only part of the story. Sleep also involves profound changes in brain chemistry. During waking hours, several neurotransmitter systems work together to maintain alertness. Acetylcholine promotes attention and learning. Norepinephrine increases arousal and vigilance. Serotonin modulates mood and sensory processing. Histamine keeps you awake and alert. And orexin, produced by neurons in the hypothalamus, acts like a master switch, stabilizing the waking state. As sleep approaches, these wake-promoting systems gradually quiet. GABA, the brain's primary inhibitory neurotransmitter, becomes increasingly active, particularly in sleep-promoting regions like the ventrolateral preoptic nucleus. This creates a mutual inhibition, a push-pull between wake-promoting and sleep-promoting systems. When sleep-promoting neurons gain the upper hand, consciousness fades. During NREM sleep, levels of acetylcholine, norepinephrine, serotonin, and histamine remain low. This neurochemical silence allows the brain to perform housekeeping functions that would be difficult or impossible during the noisy activation of wakefulness. But during REM sleep, the chemical landscape shifts dramatically. Acetylcholine surges to waking levels, or even higher, driving the intense brain activation characteristic of REM. Yet norepinephrine and serotonin fall to their lowest levels of the entire day. This unique neurochemical cocktail, high acetylcholine with minimal norepinephrine and serotonin, may explain many features of REM dreams: their vividness and emotional intensity, their bizarre and illogical narratives, and the difficulty in forming coherent memories of them. Different brain regions show different patterns of activity across sleep stages. During NREM sleep, activity decreases in many regions, particularly in the prefrontal cortex, which governs executive function, decision-making, and self-awareness. The thalamus, which normally acts as a relay station sending sensory information to the cortex, reduces its transmission during NREM, contributing to the reduced awareness of the external world. Meanwhile, some regions remain active or even increase their activity. The hippocampus, for instance, shows bursts of activity during NREM called sharp wave ripples, brief but intense activations associated with memory. Consolidation. During REM sleep, the pattern reverses in fascinating ways. Sensory and motor cortices activate strongly. The visual cortex lights up, generating the imagery of dreams. The amygdala and other limbic structures involved in emotion show high activity, possibly explaining the emotional intensity of dreams. But the prefrontal cortex, that seat of logic and self-reflection, remains relatively quiet. This may explain why we don't question the impossible events in our dreams, why we accept flying or talking animals or sudden scene changes without skepticism. The sleeping brain also shows remarkable regional coordination. During slow-wave sleep, patterns of neural activity ripple across the cortex in waves that travel from front to back, coordinating activity across distant brain regions. These traveling waves may help synchronize the various offline processing tasks that occur during sleep, ensuring that different brain regions work together even in the absence of external input. The brain during sleep is not resting. It is reorganizing, consolidating, clearing, and preparing. The patterns of electrical activity, the ebb and flow of neurotransmitters, and the coordinated activation of different brain regions all serve specific functions. Sleep transforms the brain into a state uniquely suited for maintenance, restoration, and integration of the day's experiences. The molecular machinery, adenosine and sleep pressure. Behind the scenes of sleep's architecture lies an elegant molecular mechanism that builds your need for sleep with each passing hour of wakefulness. This mechanism, known as the homeostatic sleep drive or sleep pressure, operates through a simple but powerful molecule called adenosine. Your brain is an energy-hungry organ. Though it represents only about 2% of your body weight, it consumes roughly 20% of your energy at rest. This energy comes primarily from a molecule called adenosine triphosphate, or ATP, the universal currency of cellular energy. As neurons fire, process information, form connections, and maintain their electrical potentials, they constantly break down ATP into its components. One of these breakdown products is adenosine. During wakefulness, as your brain actively processes the world around you, adenosine gradually accumulates in the extracellular space between neurons. The longer you stay awake, the more adenosine builds up, particularly in regions of the brain associated with wakefulness and attention, such as the basal forebrain. This accumulation is not merely a byproduct of neural activity. It serves as a biochemical timer, a molecular measure of how long you've been awake. According to research published in Proceedings of the National Academy of Sciences, adenosine acts through specific receptors on the surface of neurons, particularly the A1 and A2A adenosine receptors. When adenosine binds to A1 receptors on wake-promoting neurons in areas like the basal forebrain and hypothalamus, it inhibits their activity, making it harder for these neurons to fire and promoting sleepiness. Simultaneously, adenosine binding to receptors on sleep-promoting neurons in regions like the ventrolateral preoptic nucleus can enhance their activity, actively promoting sleep. This is the essence of sleep pressure. As adenosine accumulates during wakefulness, it simultaneously suppresses wake-promoting systems and activates sleep-promoting ones. The result is a growing urge to sleep, a mounting pressure that becomes increasingly difficult to resist. After about 16 hours of wakefulness, adenosine levels are high enough that most people experience strong sleepiness. After 24 hours without sleep, the pressure becomes overwhelming. But adenosine's effects extend beyond simply making you feel sleepy. Research has shown that adenosine levels influence the depth and quality of sleep itself. Higher adenosine levels at sleep onset are associated with more slow-wave sleep, that deep, restorative N3 stage. This makes biological sense. If you've been awake longer, accumulating more adenosine, your brain has more metabolic debt to repay, more restoration to accomplish, and it does this through deeper sleep. The brilliant simplicity of the adenosine system becomes even more apparent when we consider caffeine, the world's most popular stimulant. Caffeine works not by providing energy directly, but by blocking adenosine receptors. When you drink coffee or tea, caffeine molecules occupy adenosine binding sites, preventing adenosine from exerting its sleep-promoting effects. You feel more alert not because the need for sleep has disappeared, but because you've temporarily blocked the signal telling your brain you're tired. The adenosine is still there, still accumulating, but its message is being jammed. This is why, when caffeine wears off, you often experience a crash, as all that accumulated adenosine suddenly gains access to its receptors again. Recent studies using newly developed adenosine sensors have revealed the moment-to-moment dynamics of adenosine in living brains. As reported in Science, researchers found that adenosine levels fluctuate rapidly in response to neural activity, rising during periods of wakefulness and particularly during demanding cognitive tasks. But they also discovered something fascinating. Specific populations of neurons, particularly glutamatergic neurons in the basal forebrain, actively release adenosine in response to their own activity. The more these wake-related neurons fire, the more adenosine they release, creating a self-limiting mechanism. As you stay awake and these neurons work hard to keep you alert, they gradually undermine their own effectiveness by releasing the very molecule that promotes sleep. During sleep, adenosine levels decline. The processes that break down ATP continue, of course, but the overall metabolic demands of the brain decrease during sleep, particularly during NREM stages. Additionally, various mechanisms work to clear accumulated adenosine. Sleep is, in a sense, the discharge of the adenosine battery that charged during wakefulness. Individual differences in adenosine-related genes help explain why some people are more sensitive to sleep loss than others. Research on a gene called ADA, which encodes an enzyme that breaks down adenosine, has shown that people with certain variants of this gene accumulate adenosine more slowly during wakefulness and show less severe impairment during sleep deprivation. Conversely, people with variants that reduce the enzyme's activity accumulate adenosine more quickly, experience stronger sleep pressure, and show greater cognitive impairment when sleep-deprived. The adenosine system represents just one aspect of sleep's homeostatic regulation, but it's a crucial one. It provides a biochemical explanation for a universal experience. The longer you're awake, the sleepier you become. It links cellular metabolism to the behavioral need for sleep. And it offers insights into why sleep deprivation is so debilitating, and why no amount of willpower can fully overcome the drive to sleep when adenosine levels are high. The molecular machinery of sleep pressure is simple, elegant, and relentless, ensuring that eventually, no matter how hard you try to stay awake, sleep will claim you. Circadian rhythms, the body's clock. While adenosine tracks how long you've been awake, another system operates in parallel, governing when you feel sleepy and when you feel alert based not on how long you've been awake, but on what time of day it is. This is the circadian system, your body's internal clock, and it exerts profound influence over sleep and nearly every other aspect of physiology. The word circadian comes from the Latin circa, meaning about, and dies, meaning day. Circadian rhythms are approximately 24-hour cycles in biological processes that persist even in the absence of external time cues. If you were placed in a dark cave with no clocks, no sunrise or sunset, no external indicators of time, your body would still maintain a roughly 24-hour rhythm of sleepiness and alertness, of body temperature fluctuations, of hormone release, and of countless other physiological processes. The master circadian pacemaker in mammals resides in a tiny region of the brain called the suprachiasmatic nucleus, or SCN, located in the hypothalamus just above the optic chiasm, where the optic nerves cross. The SCN contains only about 20,000 neurons in humans, yet these cells generate one of the most fundamental rhythms in biology. Individual neurons in the SCN contain molecular clocks, feedback loops of gene expression that take approximately 24 hours to complete one cycle. Proteins encoded by clock genes accumulate during the day, then activate genes that suppress their own production, creating an oscillation that repeats day after day. But the SCN does more than just maintain its own rhythm. It broadcasts timing information throughout the brain and body, synchronizing peripheral clocks in virtually every organ and tissue. The liver has its own clock, regulating metabolism. The kidneys have clocks that affect urine production. Even individual cells in your skin maintain circadian rhythms. The SCN coordinates all these distributed clocks, ensuring that the body's myriad processes unfold in proper sequence. Perhaps the most important function of the circadian system regarding sleep is its influence on melatonin, often called the hormone of darkness. Melatonin is produced by the pineal gland, a small structure deep in the brain. The SCN controls melatonin production through a multi-step pathway involving the sympathetic nervous system. According to research published in Psychiatric Times, during daylight hours, The SCN actively suppresses melatonin production. But as evening approaches and light levels fall, this suppression is released. Melatonin synthesis begins, and its levels in the bloodstream rise, typically reaching their peak between 3 and 4 m before declining again toward dawn. Melatonin is sometimes misunderstood as a sleeping pill, but its role is more subtle. Melatonin's primary function is as a signal of darkness, conveying information about the light-dark cycle to the rest of the body and brain. As described in research from the British Journal of Pharmacology, melatonin acts through two receptor subtypes, MT1 and MT2, which are densely expressed in the SCN itself and in other brain regions. The rise of melatonin opens what sleep researchers call the sleep gate, a window of time when sleep becomes physiologically easier to initiate. About 2 hours before your regular bedtime, as melatonin begins to rise, sleep propensity increases sharply. Before this rise, initiating sleep is difficult. After it, sleep comes much more easily. But melatonin doesn't simply make you sleepy, it also helps set the timing of the circadian clock itself, acting as a feedback signal. When melatonin binds to receptors in the SCN, it can shift the phase of the circadian rhythm, helping to synchronize the internal clock with the external world. This is why melatonin supplementation can help with jet lag or shift work, not primarily because it makes you sleepy, but because it helps reset your circadian clock to the new time zone. The circadian influence on sleep is most apparent in the interaction between the circadian drive for wakefulness and the homeostatic sleep pressure from adenosine. During a typical day, these two systems work in opposition. As you progress through the day and adenosine builds up, you might expect to feel progressively sleepier. But you don't, at least not until evening. This is because the circadian drive for wakefulness also increases through the afternoon, counteracting rising sleep pressure. It's only in the evening, when both melatonin rises and the circadian wake drive diminishes, that sleepiness becomes overwhelming. This opponent-process arrangement, where circadian and homeostatic systems push in opposite directions during the day, allows for consolidated wakefulness and consolidated sleep. Without it, you would feel increasingly tired throughout the day and might take frequent naps. The circadian system essentially provides a boost of alertness that peaks in the early evening, just when homeostatic sleep pressure is highest after a full day awake. This wake maintenance zone, as it's sometimes called, keeps you alert through the evening hours. But once it releases and melatonin rises, sleepiness comes on quickly and powerfully. Light is the primary synchronizer of circadian rhythms, and the pathway by which light influences the clock is direct and specific. A subset of retinal ganglion cells in the eye contain a photopigment called melanopsin, which is maximally sensitive to blue light. These cells send signals directly to the SCN, providing information about ambient light levels. Bright light, especially in the blue wavelengths prevalent in daylight and in modern screens, strongly suppresses melatonin and shifts circadian phase. Even moderate room lighting can reduce melatonin secretion, which is why exposure to bright lights in the evening can make it harder to fall asleep on time. Your circadian rhythm is not set in stone. It can be shifted earlier or later depending on when you're exposed to light. Bright light in the morning shifts the clock earlier, promoting earlier sleep and wake times. Bright light in the evening delays the clock, pushing sleep and wake times later. This flexibility allows the circadian system to adapt to seasonal changes in day length and to geographical moves across time zones. But it also makes the system vulnerable to disruption in modern life, where artificial lighting extends effective day length and electronic devices emit large amounts of blue light late into the evening. Individual differences in circadian rhythms, often called chronotypes, are substantial and partly genetic. Some people are natural early birds or larks, whose circadian clocks run slightly fast, making them want to sleep and wake early. Others are night owls, with slower running clocks that prefer later schedules. These differences are real and biological, not merely matters of discipline or preference. Trying to force a strong night owl to consistently wake at 6 a.m. is fighting against their biological clock, and the reverse is true for larks forced into late schedules. The circadian system is ancient and fundamental, regulating not just sleep, but metabolism, immune function, cognitive performance, and even cell division throughout the body. When this system falls out of sync with the external world, whether due to shift work, jet lag, or simply chronic sleep restriction with late nights and early mornings, the consequences extend far beyond simple sleepiness. Circadian disruption increases risk for metabolic disorders, cardiovascular disease, mood disorders, and even cancer. The timing of sleep matters as much as its duration. Sleeping during your biological night, when melatonin is high and your circadian clock expects sleep, is more restorative than sleeping for the same duration at the wrong circadian time. The circadian system is the conductor of the body's symphony, ensuring that all the instruments play not just correctly, but at the right time, in the right order, in harmony with the turning of the earth. Why we sleep. The functions. For all that science has revealed about the mechanisms of sleep, its stages, its molecules, and its rhythms, perhaps the most fundamental question remains the simplest and most difficult to answer. Why do we sleep? What function does this nightly disappearance from consciousness serve? The answer, it turns out, is not singular, but plural. Sleep serves multiple vital functions, and we're only beginning to understand their full scope. One of the most well-established functions of sleep is its role in memory consolidation, the process by which new memories are stabilized and integrated with existing knowledge. The story of sleep's role in memory emerged gradually through decades of research, but the evidence is now overwhelming. As reported in Nature Neuroscience, both NREM and REM sleep appear critical for different aspects of memory processing. During waking hours, you encounter vast amounts of information. You meet new people, learn new facts, have new experiences. These new memories initially depend heavily on the hippocampus, a brain structure that can rapidly encode new information but has limited storage capacity. Without consolidation, these hippocampus-dependent memories would be fragile and easily lost. Sleep provides a unique opportunity for consolidation because the brain is freed from processing new incoming information and can focus on organizing and strengthening what has already been learned. The consolidation process involves a dialogue between the hippocampus and the neocortex, the outer layer of the brain responsible for long-term storage of knowledge. During slow-wave sleep, the hippocampus replays patterns of neural activity that occurred during learning. These reactivations trigger corresponding changes in the neocortex, gradually transferring memories from the temporary hippocampal store to more permanent neocortical networks. This transfer makes memories less dependent on the hippocampus and more resistant to interference. Brain imaging studies in humans have directly observed this process. When people learn a spatial task during the day and then sleep, brain scans during subsequent slow-wave sleep show coordinated activation in both the hippocampus and regions of the cortex that were active during initial learning. The amount of this reactivation during sleep predicts how well the memory will be retained. Artificially enhancing slow oscillations during sleep, using techniques like transcranial stimulation, improves memory. Consolidation, while disrupting slow-wave sleep, impairs it. But memory consolidation during sleep goes beyond simple strengthening. Sleep also appears to extract the gist or schema from experiences, distilling general rules and patterns from specific episodes. You might remember the core message of a conversation or the general layout of a new building while forgetting specific details. This abstraction and integration of new information with existing knowledge structures appears to be a special function of sleep. The brain isn't just preserving memories during sleep, it's actively transforming them, finding connections, extracting meaning. Different types of memory appear to benefit from different sleep stages. Declarative memories, facts and events that you can consciously recall, benefit particularly from slow-wave sleep. Procedural memories, skills and habits like riding a bike or playing an instrument, show enhanced consolidation during both slow-wave sleep and REM sleep. Emotional memories, events with strong emotional content, also show enhanced consolidation, and this process appears linked to REM sleep and the high activity of the amygdala during this stage. Beyond memory, sleep serves critical functions in cellular and metabolic regulation. During deep sleep, growth hormone is released, promoting tissue repair, muscle growth, and bone development, a relationship established through decades of endocrinological research. The immune system becomes particularly active during sleep, producing infection-fighting substances and mounting enhanced responses to pathogens. The evidence for this is compelling. Controlled studies have shown that people who are sleep-deprived demonstrate reduced immune function and are more susceptible to infections when experimentally exposed to viruses. This observation helps explain why you often feel sleepy when sick. Based on what we understand about immune-sleep interactions, researchers interpret this as your immune system ramping up its activity, with sleep helping it work more effectively. Sleep also plays vital roles in metabolic regulation. Hormones that control hunger and satiety, leptin and ghrelin, show circadian variation and demonstrate sensitivity to sleep loss in experimental studies. Sleep deprivation consistently increases ghrelin, which stimulates appetite, and decreases leptin, which signals fullness, creating a measurable drive to eat more. This helps explain the well-established epidemiological link between insufficient sleep and weight gain. Sleep also affects insulin sensitivity and glucose metabolism in ways that have been documented through careful clinical studies. Even a few nights of partial sleep restriction can reduce the body's ability to regulate blood sugar, temporarily pushing healthy individuals toward a pre-diabetic state, an effect that reverses with sleep recovery. Emotional regulation represents another crucial function of sleep, particularly REM sleep. During waking hours, emotional experiences activate the amygdala, the brain's emotional processing center. If these emotions are very intense, they can trigger stress responses that are useful in the moment, but potentially harmful if prolonged. Sleep, especially REM sleep, appears to help process emotional experiences, though the precise mechanisms remain an active area of investigation. The interpretation that sleep reduces emotional charge while preserving memory of the event draws support from brain imaging studies showing that when people are sleep-deprived, their amygdala shows exaggerated responses to negative stimuli, and connectivity between the amygdala and the prefrontal cortex, which normally regulates emotional responses, is measurably reduced. Sleep-deprived people consistently show greater emotional reactivity, irritability, and difficulty controlling their responses across multiple experimental paradigms. Sleep may also facilitate creativity and problem-solving, though this relationship is more complex and context-dependent than popular accounts sometimes suggest. Controlled laboratory studies have demonstrated that after learning a problem, people who sleep before attempting to solve it show higher rates of discovering shortcuts or novel solutions than people who stay awake for an equivalent period. Based on converging evidence from multiple research groups, this could relate to the way sleep allows new information to be integrated with existing knowledge, potentially creating connections that weren't obvious during focused waking thought, though alternative explanations involving reduced mental fatigue haven't been entirely ruled out. Perhaps most intriguingly, recent research has revealed that sleep serves a unique housekeeping function, clearing metabolic waste from the brain. This function, mediated by the glymphatic system, is so important it deserves its own section. But it underscores a crucial point. The brain cannot maintain itself during waking activity alone. Sleep provides a distinct physiological state required for basic maintenance. The functions of sleep are diverse, interacting, and essential. Memory consolidation, immune enhancement, metabolic regulation, emotional processing, creative insight, waste clearance. Each of these functions has been demonstrated in careful studies, and likely many more remain to be discovered. The question is not whether sleep serves important functions, but whether we've yet understood the full extent of its importance. What's certain is that these functions cannot be performed adequately during wakefulness or through any shortcut or substitute. Sleep is not a luxury or an evolutionary accident. It is a fundamental biological necessity, as important to health and well-being as food, water, or air. The glymphatic system, the brain's cleaning crew. In 2012, neuroscientists made a discovery that would fundamentally change our understanding of why sleep is necessary. Working with mice at the University of Rochester, researcher Maiken Nedergaard and her team identified a system, previously unknown, that clears waste products from the brain, and they found that this system operates primarily during sleep. They called it the glymphatic system, a name that combines glial cells, the brain's support cells, with lymphatic, referencing its similarity to the body's lymphatic system that drains waste from other tissues. The existence of the glymphatic system addresses a long-standing puzzle in neuroscience. The brain, despite representing only 2% of body weight and consuming 20% of the body's energy, lacks a traditional lymphatic system for waste removal. For decades, scientists assumed that waste products simply diffused away through the dense brain tissue, but mathematical models suggested the rates of diffusion were far too slow to account for the efficient clearance of metabolic waste that must occur to keep the brain healthy. The glymphatic discovery revealed a much more sophisticated system. According to research published in Science, the system works through a precisely orchestrated process. Cerebrospinal fluid, or CSF, the clear liquid that surrounds the brain and spinal cord, flows into the brain along the outside of arteries, through specialized channels called perivascular spaces. Think of it as a plumbing system running alongside the blood vessels. This inflowing CSF then enters the brain tissue itself through water channels called aquaporin 4, or AQP4, which are densely expressed on the end feet of astrocytes, star-shaped glial cells that wrap around blood vessels like biological sleeves. Once inside the brain tissue, the CSF mixes with interstitial fluid, the liquid that fills the microscopic spaces between cells. This mixing creates a flow that sweeps through the tissue like a gentle tide, collecting waste products, proteins that have misfolded or aggregated, and metabolic byproducts like adenosine. The contaminated fluid then drains out of the brain along the outside of veins, again through perivascular spaces, ultimately reaching the lymphatic vessels in the neck, where it joins the body's general circulation and is processed by the liver and kidneys. What makes this system remarkable is not just its existence, but its regulation by sleep state. Research published in Science demonstrated that the glymphatic system is dramatically more active during sleep than during wakefulness, clearing beta amyloid, a protein implicated in Alzheimer's disease, at approximately twice the rate during sleep compared to waking. During sleep, the extracellular space in the brain expands by up to 60%, allowing for greater flow of fluid and more efficient waste clearance, a finding that initially surprised researchers given that brain volume remains constant. This expansion appears to be controlled by the neurotransmitter norepinephrine. During wakefulness, norepinephrine levels are high, and the extracellular space is relatively constricted. During sleep, especially during slow-wave sleep, norepinephrine levels drop, the extracellular space expands, and glymphatic clearance accelerates dramatically. The waste products cleared by the glymphatic system are not trivial. They include amyloid beta, a protein that accumulates in plaques in Alzheimer's disease, and tau, another protein implicated in neurodegeneration. Studies using PET imaging have shown that even a single night of sleep deprivation can increase amyloid beta levels in the brain by 5 to 10%, as measured in the hippocampus and thalamus of healthy young adults. Based on longitudinal epidemiological data, chronic sleep disruption over years may allow these proteins to accumulate to levels that could contribute to dementia risk, though this interpretation remains an area of active investigation. The observed association between sleep disorders, particularly sleep apnea with its repeated breathing interruptions, and increased Alzheimer's risk may be mediated partly through impaired glymphatic clearance, a hypothesis supported by animal studies, but not yet proven in humans. Recent research, including a landmark 2024 study published by Oregon Health and Science University, has provided direct evidence of the glymphatic system in humans. Using specialized imaging techniques during neurosurgery, researchers visualized the perivascular spaces and the flow of contrast agent through them, confirming that the structural elements of the glymphatic system exist in human brains and function as predicted from animal studies. The glymphatic system's dependence on sleep helps explain a fundamental mystery. Why do we need to be unconscious to clean our brains? Why can't this happen during quiet waking? The answer appears to be that the neurochemical state of wakefulness, with high norepinephrine and active neural processing, prevents the expansion of extracellular space necessary for efficient glymphatic flow. The brain must choose between processing information and clearing waste. It cannot do both simultaneously at high efficiency. Sleep, particularly slow-wave sleep with its characteristic slow oscillations and reduced norepinephrine, provides the optimal state for waste clearance. This discovery has profound implications for understanding sleep disorders and sleep deprivation. When you don't get enough sleep, or when sleep quality is poor, your brain may not have adequate time in the optimal state for waste clearance. Metabolic waste and protein aggregates may accumulate. Over time, this accumulation could contribute to cognitive decline, mood disorders, and neurodegenerative diseases. The glymphatic system provides a mechanistic link between poor sleep and long-term brain health. Several factors can affect glymphatic function. Sleep position matters. Sleeping on your side appears to enhance glymphatic clearance compared to sleeping on your back or stomach. Aging reduces glymphatic efficiency, which may be one reason older adults are more vulnerable to neurodegenerative diseases even without obvious sleep disorders. Alcohol, despite its sedative effects, disrupts sleep architecture and may impair glymphatic function. Cardiovascular health affects glymphatic flow, since the pulsations of arteries help drive the movement of CSF. The glymphatic system represents a fundamental function of sleep: maintenance. Just as your body needs sleep to repair tissues, to consolidate memories, to regulate metabolism, it needs sleep to literally clean the brain. This cleanup cannot wait. It must happen regularly, and it can only happen effectively during sleep. The discovery of the glymphatic system elevates sleep from merely beneficial to absolutely essential for long-term brain health. Every night you sleep, your brain rinses itself clean, preparing for another day of thought, perception, and experience. Skip or skimp on that nightly cleaning session, and the consequences accumulate. Silently, but relentlessly. Memory consolidation during sleep. We've touched on memory consolidation as one function of sleep, but this process is so important and so well studied that it deserves deeper exploration. The transformation of experiences into lasting memories is one of the most remarkable things that happens while you sleep, a process that reveals how sleep actively shapes who you are and what you know. Memory consolidation is not a simple stamping process that fixes experiences in place like photographs in an album. Rather, it's an active process of reorganization and integration. When you learn something new, say a list of words, a route through a city, or how to play a musical phrase, the initial memory trace is formed in the hippocampus. This structure, located deep in the temporal lobes, has the remarkable ability to rapidly bind together diverse elements of an experience, the sights, sounds, emotions, and context, into a coherent memory. But the hippocampus has limited capacity. Without consolidation, new memories would overwrite old ones, and knowledge would be fleeting. Consolidation solves this problem through a dialogue between the hippocampus and the neocortex. During sleep, particularly during slow-wave sleep, the hippocampus replays recent memories. This replay is not random. As research published in Nature Neuroscience has shown, sequences of hippocampal neurons fire in the same order they fired during the original experience, but compressed in time, often occurring 10 to 20 times faster than the original event. These accelerated replays occur during sharp wave ripples, brief but intense bursts of high-frequency activity that sweep through the hippocampus during slow-wave sleep. These hippocampal replays are coordinated with activity in the neocortex through the slow oscillations that characterize deep sleep. The slow oscillation, a wave of neural activity that travels across the cortex about once per second, appears to timestamp moments when the cortex is receptive to input from the hippocampus. During the up phase of the slow oscillation, cortical neurons are more excitable and likely to fire. Hippocampal replays tend to occur during these up phases, ensuring that the message gets through. Additionally, thalamic sleep spindles, those brief bursts of 11 to 16 Hz activity, often occur during the same up phases, and spindle activity may help strengthen the synaptic connections that encode the replayed memory in the cortex. This three-way coordination, slow oscillations providing windows of opportunity, hippocampal replays transmitting memory content, and spindles strengthening cortical connections, represents an elegant mechanism for memory transfer. Research has shown that manipulating any of these elements affects consolidation. Artificially enhancing slow oscillations improves memory. Disrupting spindles impairs it, and the degree of hippocampal cortical coordination during sleep predicts memory retention. The outcome of this process is a memory that has been transformed. What began as a hippocampus-dependent episodic memory, a specific event tied to a particular time and place, gradually becomes a neocortical semantic memory, a piece of knowledge stripped of some contextual details, but integrated with existing understanding. You might remember learning a historical fact without remembering exactly when or where you learned it. The fact has been abstracted and incorporated into your broader knowledge of history. But consolidation isn't just about transferring memories. Sleep also appears to selectively strengthen some memories while weakening others. Memories that are emotionally important, that have been tagged as significant, or that fit with existing schemas are preferentially consolidated. Trivial or redundant information may be allowed to fade. This selective process means that sleep doesn't just preserve, it edits, prioritizing what matters. REM sleep also plays a role in consolidation, though its specific functions are still debated. Some evidence suggests that REM sleep is particularly important for consolidating procedural memories, skills, and habits. Learning a motor skill like a piano piece or a tennis serve often shows improvement after a night's sleep, a phenomenon called sleep-dependent motor memory consolidation. This improvement is associated with increased REM sleep and with changes in brain regions involved in motor control. REM may also play a special role in consolidating emotional aspects of memories, integrating them in ways that reduce their emotional charge while preserving their informational content. The emotional processing during REM may explain why nightmares can be so frequent after traumatic events, and why conditions like PTSD, where emotional memories resist integration, are often accompanied by sleep disturbances. The normal process by which REM sleep helps process and contextualize emotional memories may be disrupted, leading to intrusive memories and hyperarousal. Targeted memory reactivation studies provide some of the most direct evidence that specific memories are processed during sleep. In these experiments, people learn associations between images or locations and specific sounds. During subsequent sleep, those sounds are played quietly. Brain imaging reveals reactivation of the brain regions involved in encoding those memories, and this reactivation leads to improved memory performance. The brain is literally rehearsing specific memories during sleep in response to subtle cues. The memory consolidation functions of sleep have practical implications. Students who pull all-nighters before exams are fighting against their own biology. The information crammed the night before won't be consolidated effectively without sleep, and performance will suffer not just from sleepiness, but from inadequate memory processing. Conversely, strategic use of sleep after learning, even a brief nap, can substantially enhance retention. Sleep's role in memory is so fundamental that some researchers have proposed it may be the primary reason sleep evolved in the first place. The argument goes that as brains became larger and more complex, they needed a distinct state for offline processing of experiences. Wakefulness is for encoding new information. Sleep is for organizing and integrating it. Without sleep, memories would remain unstable, unorganized, isolated from existing knowledge. Learning would be inefficient, and knowledge would be fragile. Every night, as you sleep, your brain is not merely resting. It is weaving today's experiences into the fabric of all you know, building the narrative of your life, constructing the self that will wake in the morning slightly changed, slightly wiser, carrying forward the essence of what you learned and discarding the rest. Dreams, the science behind the stories. Dreams are among the most mysterious and captivating experiences of sleep. For much of human history, dreams were attributed to supernatural sources, messages from gods, visits from spirits, or glimpses of alternate realities. Modern neuroscience has brought dreams firmly into the realm of biology, revealing them as products of brain activity during sleep. Yet even with this understanding, dreams retain their strangeness and fascination. Why does the sleeping brain generate these elaborate, often bizarre narratives? What function, if any, do they serve? First, let's clarify when dreaming occurs. For decades, it was believed that dreams happened exclusively during REM sleep, that stage characterized by rapid eye movements, brain activity similar to wakefulness, and muscle paralysis. And indeed, REM sleep is associated with the most vivid, story-like, hallucinatory dreams. If you wake someone during REM sleep, they will report a dream about 80% of the time, and these dreams tend to be elaborate, emotional, visually vivid, and often bizarre or illogical. But research has shown that dreaming is not exclusive to REM sleep. People awakened from NREM sleep also report mental experiences, though these tend to be different in character. According to studies published in PMC, NREM dreams are typically shorter, less vivid, more thought-like, and more grounded in reality. They might involve replaying events from the day, thinking about upcoming tasks, or experiencing fragmented images rather than continuous narratives. the difference is not absolute. Some NREM dreams can be quite vivid, especially later in the night, and some REM dreams are mundane. But on average, the two types of sleep produce different dream experiences. Why do dreams during REM sleep have their characteristic features? The neurochemistry and neural activation patterns of REM provide clues. During REM sleep, as we've discussed, acetylcholine levels are high, driving intense brain activation. Visual cortex is strongly active, generating the imagery of dreams. The limbic system, including the amygdala and anterior cingulate cortex, shows high activity, contributing to the emotional intensity of dreams. But critically, the dorsolateral prefrontal cortex, which governs executive functions like logic, self-reflection, and reality testing, is relatively quiet. This pattern of activation, high visual and emotional areas, low prefrontal, may explain why dreams are so visually vivid and emotionally charged, yet so uncritical. In dreams, you rarely question impossibilities. You fly without wondering how. You meet people who are dead without realizing the contradiction. The scene changes abruptly, and you don't notice. Your prefrontal cortex, which during waking would immediately flag these as impossible or illogical, is offline. The neurochemical environment of REM, particularly the near-complete absence of norepinephrine and serotonin, may also contribute to dream bizarreness and the difficulty in forming stable memories of dreams. These neurotransmitters are involved in attention, working memory, and logical thought. Their absence may make the dream state more fluid, more associative, less constrained by the rules that govern waking thought. But why do we dream at all? Here we enter territory where scientific certainty gives way to competing interpretations, each supported by evidence, but none yet proven definitive. Multiple theories have been proposed, and researchers continue to debate their relative merits. Freud famously proposed that dreams represent wish fulfillment, expressing unconscious desires in disguised form. While psychoanalytic dream interpretation has fallen largely out of favor in neuroscience, the broader idea that dreams carry psychological significance hasn't been entirely abandoned. Based on patterns observed in dream content studies, some modern researchers suggest that dreams may reflect our concerns, process emotions, or work through problems, though likely not in the symbolic manner Freud envisioned. The threat simulation theory, proposed by cognitive neuroscientist Antti Revonsuo, offers an evolutionary interpretation. Dreaming, this hypothesis suggests, may have evolved as a way to rehearse responses to dangerous situations. The evidence supporting this includes consistent findings that dreams frequently contain threatening elements, being chased, falling, confronting dangers. The interpretation draws on the observation that people who've experienced trauma often have dreams with even more threatening content, which could imply an adaptive increase in threat rehearsal, though alternative explanations exist. The emotional regulation theory focuses on memory and mood. According to this framework, dreams during REM sleep may help process emotional experiences from waking life. The reasoning goes like this. By replaying emotional events during sleep in the unique neurochemical environment of REM, with high acetylcholine but low norepinephrine, the brain might be able to extract the informational content of emotional memories while reducing their emotional intensity. Some researchers call this emotional memory triage. Based on brain imaging and behavioral studies, this could explain why, after sleep, you often remember what happened but feel less intensely about it, though the precise mechanisms remain under investigation. The activation synthesis hypothesis, one of the earliest neurobiological theories of dreaming, proposed that dreams result from the brain's attempt to make sense of neural activity during REM sleep. The brainstem generates bursts of activity that activate various cortical regions, and the cortex attempts to synthesize these activations into coherent narratives. This theory has evolved considerably since its initial formulation. Most researchers no longer characterize the activations as entirely random, but the core principle that dreams reflect the brain's drive to create meaning from internal activity continues to influence modern interpretations. More recent theoretical frameworks emphasize dreams' potential role in creative thinking and problem-solving. The loose associations, the bizarre combinations, the suspension of logical constraints that characterize dreams may, according to this view, allow the brain to make unexpected connections and see problems from novel angles. Famous anecdotes exist of scientific and artistic insights occurring in dreams, from Kekule's dream of a snake biting its tail, which reportedly led to understanding the ring structure of benzene, to Paul McCartney claiming to have dreamed the melody of Yesterday. While such anecdotes must be interpreted cautiously, controlled laboratory studies have demonstrated that REM sleep does enhance performance on creative problem-solving tasks, and that people are more likely to discover hidden rules or insights after sleep, lending empirical support to the creativity hypothesis. The phenomenon of lucid dreaming, where the dreamer becomes aware they're dreaming and can sometimes exert control over the dream, provides a unique window into dream mechanisms. Lucid dreams most commonly occur during REM sleep, often in the later cycles of the night. Neuroimaging studies of lucid dreamers have shown increased activity in the prefrontal cortex compared to non-lucid REM sleep, suggesting that lucidity involves a partial reactivation of the self-reflective capacities that are normally suppressed during dreaming. Why are dreams so hard to remember? The neurochemistry of REM sleep, particularly the low levels of norepinephrine, may impair the encoding of memories during the dream itself. Additionally, the transition from sleep to wakefulness involves a dramatic neurochemical shift that may disrupt fragile dream memories. Most dreams are forgotten within minutes of waking unless you actively rehearse them. This forgetting might actually be adaptive, preventing confusion between dream events and real experiences. The content of dreams reflects both universal themes and individual experiences. Across cultures, common dream themes include falling, being chased, flying, showing up unprepared for an exam, and teeth falling out. These universal themes may reflect shared human concerns and experiences, but dreams also incorporate specific elements from your waking life, your recent experiences, your preoccupations, your social relationships. Dreams appear to reflect your brain processing and integrating waking experiences, even if in ways that seem nonsensical. Despite decades of research, dreams remain somewhat mysterious. We know where they come from in terms of brain activity. We have good theories about some of their functions, but we don't have a complete explanation for their often bizarre content or their subjective intensity. Perhaps dreams serve multiple functions simultaneously, consolidating memories, processing emotions, simulating threats, facilitating creativity. Perhaps they're a byproduct of processes that serve other functions, or perhaps there's a primary function we haven't yet identified. What's clear is that the dreaming brain is not idle. It's active, creative, emotional, and in its own strange way, meaningful. Every night, your brain tells you stories, vivid narratives that dissolve with dawn, leaving only fragments and feelings, whispers of another world that lives only in sleep. Sleep deprivation, when rest becomes debt. To understand how essential sleep is, we need only look at what happens when we don't get enough of it. Sleep deprivation, whether acute, a single night without sleep, or chronic, consistent, insufficient sleep over days, weeks, or years, produces widespread effects on brain and body that underscore just how fundamental sleep is to human functioning. The cognitive effects of sleep deprivation are perhaps the most immediately noticeable and the most thoroughly studied. According to research published in Neuroscience's journal, a reduction in sleep does not occur independently, but cascades through multiple cognitive domains: memory, attention, alertness, judgment, decision-making, and overall cognitive performance all decline. Attention and vigilance are particularly vulnerable. Studies using the psychomotor vigilance task, a simple test where subjects press a button as quickly as possible when a light appears, show dramatic impairments after sleep deprivation. Reaction times slow, and lapses occur where the person fails to respond at all, as if momentarily dropping into micro-sleeps. These lapses increase with continued wakefulness. After 24 hours without sleep, performance drops to levels comparable to legal intoxication. After multiple days of even partial sleep restriction, getting only 4 to 6 hours per night, cumulative deficits build up that equal the impairment of total sleep deprivation. Memory is also severely affected. Sleep deprivation impairs both the encoding of new memories during wakefulness and the consolidation that would normally occur during subsequent sleep. Students who cram all night retain less information than those who study the same amount of time, but then sleep. The memories that do form during sleep deprivation are more prone to errors and false memories. You might remember something happening that didn't, or misremember details, because your brain was too impaired to encode the information accurately. Higher-level cognitive functions show deficits as well. Decision-making becomes more rigid, less adaptive. Creativity and divergent thinking, the ability to generate novel solutions, decline. Problem-solving becomes more difficult. Interestingly, routine tasks that don't require much thought or flexibility are relatively spared. You can still perform familiar actions through habit, but anything requiring innovation, adaptation, or careful attention suffers. The emotional and psychological effects of sleep deprivation are equally concerning. Functional brain imaging has revealed that sleep deprivation amplifies activity in the amygdala, the emotional center, in response to negative stimuli. Simultaneously, connectivity between the amygdala and the prefrontal cortex, which normally regulates emotional responses, is reduced. The result is emotional dysregulation, heightened reactivity to negative events, increased irritability, reduced ability to control emotional responses, and greater susceptibility to stress. Chronic sleep deprivation increases risk for mood disorders. The relationship between sleep and depression is complex and bidirectional. Poor sleep can contribute to depression, and depression disrupts sleep, but there's no doubt they're intertwined. Similarly, anxiety and sleep problems feed each other. Sleep deprivation produces a state of hyperarousal, making it harder to relax and fall asleep even when you finally have the opportunity. The physical health consequences of insufficient sleep are wide-ranging and serious. The cardiovascular system is affected. Blood pressure rises, heart rate variability decreases, and inflammatory markers increase. Chronic short sleep is associated with increased risk of hypertension, heart disease, and stroke. The metabolic system also suffers. As mentioned earlier, sleep deprivation affects the hormones that regulate hunger and satiety, leading to increased appetite and preference for high-calorie foods. It also reduces insulin sensitivity, impairing glucose regulation and increasing diabetes risk. The immune system shows clear impairments with sleep loss. Studies have demonstrated that people who are sleep-deprived are more likely to catch a cold when exposed to the virus. Vaccination responses are reduced in sleep-deprived individuals, producing fewer antibodies. During sleep, the immune system releases cytokines that help fight infection and inflammation. Without adequate sleep, this nighttime boost is diminished. Perhaps most alarming are the effects on the glymphatic system and brain health. As discussed earlier, The glymphatic system clears metabolic waste from the brain primarily during sleep. Sleep deprivation reduces this clearance, allowing waste products like amyloid beta to accumulate. Studies using PT imaging have shown that even a single night without sleep increases amyloid beta levels in the brain. Over years or decades, chronic insufficient sleep may contribute to neurodegenerative disease risk. The effects of sleep deprivation are not uniform across individuals. Genetics play a role. Some people are more resilient to sleep loss than others, maintaining better performance with less sleep. But importantly, people are generally poor judges of their own impairment. After several days of restricted sleep, subjective sleepiness may level off even while objective performance continues to decline. You may feel like you've adapted to less sleep, but measures of cognitive performance tell a different story. The impairment is real, even if you've stopped noticing it. Can you recover from sleep deprivation? Yes, but recovery is not instant or complete. After acute total sleep deprivation, the first recovery night typically shows increased sleep depth and longer duration, with more time in slow-wave sleep and REM sleep, a phenomenon called sleep rebound. But a single recovery night may not fully restore all functions, especially higher cognitive abilities. After chronic partial sleep restriction, recovery takes longer. Several nights of extended sleep may be needed to return to baseline functioning. There's also evidence that some effects of chronic sleep deprivation may be long-lasting. The full extent of recovery from prolonged insufficient sleep, the kind many people experience across years of demanding work schedules or chronic insomnia, is still not completely understood. The brain and body are remarkably resilient, but chronic stressors leave marks. The best approach is prevention, prioritizing adequate sleep consistently rather than accruing sleep debt that requires extended recovery. The message from sleep deprivation research is clear and concerning. Sleep is not optional. It cannot be indefinitely postponed without serious consequences. The modern world, with its 24-hour lighting, electronic devices, demanding schedules, and cultural valorization of minimal sleep, creates conditions that work against our biological need for rest. Understanding the consequences of insufficient sleep isn't just academic. It's a public health imperative. When we skimp on sleep, we pay for it in cognitive performance, emotional stability, physical health, and ultimately in quality and perhaps length of life. Sleep across the lifespan. Sleep is not static across the human lifespan. From infancy through old age, sleep changes in duration, architecture, timing, and function. Understanding these changes helps illuminate both what sleep does and how to optimize it at different life stages. Newborns sleep approximately 16 to 18 hours per day, though this sleep is distributed across the entire 24-hour period in short bouts lasting 2 to 4 hours. Newborn sleep differs markedly from adult sleep in several ways. According to research published in StatPearls, newborns have 3 types of sleep: quiet sleep, similar to NREM, active sleep, similar to REM, and indeterminate sleep, which doesn't fit clearly into either category. Newborns enter sleep through REM, not NREM as adults do, and REM comprises about 50% of their sleep time. Circadian rhythms haven't yet developed, which is why newborns wake frequently around the clock, to the exhaustion of new parents. Between 2 and 3 months of age, critical developmental changes occur. Circadian rhythms begin to establish themselves, driven by the maturation of the suprachiasmatic nucleus and the onset of rhythmic melatonin secretion. Sleep onset begins to shift from REM to NREM. The total sleep cycle shortens from 50 to 60 minutes toward the adult pattern of 90 to 120 minutes. Sleep becomes more consolidated at night, with longer wake periods during the day. By 6 months, most infants sleep 6 hours continuously at night. By 12 months, they typically sleep 14 to 15 hours total per day, with most sleep occurring at night and 1 to 2 daytime naps. The proportion of REM sleep decreases toward the adult level of 20 to 25%. Childhood sees continued evolution of sleep patterns. Between ages 2 and 5, total sleep decreases from about 13 to 11 hours, with most children giving up afternoon naps. Slow-wave sleep, that deep N3 stage, is abundant in childhood, accounting for a larger proportion of sleep than in adults. This makes sense given the rapid physical and neural development occurring during these years. Children also begin to develop circadian preferences. Some become early birds, some night owls, tendencies that will persist through life. Adolescence brings dramatic changes in sleep, driven by both biological and social factors. Puberty triggers a shift in circadian timing, a delay in the release of melatonin that makes teenagers naturally inclined to stay up later and sleep later. The optimal sleep schedule for most teenagers would be sleeping from midnight or later until 9 or 10 am. But social demands, early school start times, homework, extracurricular activities create chronic sleep restriction. Most teenagers don't get the 8 to 10 hours they need. Slow-wave sleep begins to decline during adolescence, a trend that continues through life. REM sleep latency, the time from sleep onset to the first REM period, also decreases compared to childhood. Adult sleep, ideally, stabilizes into a pattern of 7 to 9 hours per night with consistent bed and wake times. But individual variation is substantial. Some adults genuinely need only 6 hours, while others require 10, differences driven partly by genetics. The proportion of time in different sleep stages remains relatively consistent through early and middle adulthood. But even in healthy young adults, lifestyle factors like work schedules, light exposure, caffeine use, and stress can dramatically affect sleep quality and quantity. Aging brings further changes, some of which are problematic. Older adults often have difficulty maintaining sleep, with more frequent awakenings during the night and earlier morning waking. Slow-wave sleep continues its decline, with many elderly people getting little to no N3 sleep. REM sleep also decreases slightly. Total sleep time may decrease, though sleep need doesn't necessarily change. Older adults may need the same duration, but have more difficulty achieving it. These changes reflect alterations in multiple systems. The circadian pacemaker shows reduced amplitude, producing less robust sleep-wake rhythms. Melatonin secretion decreases with age. The homeostatic sleep drive, the buildup of sleep pressure from adenosine, may weaken. Medical conditions and medications common in older age can further disrupt sleep. Sleep disorders like sleep apnea and restless legs syndrome become more prevalent. But not all age-related sleep changes are inevitable. Much of the sleep disruption in older adults results from correctable factors: poor sleep habits, inadequate light exposure during the day, medical issues that can be treated, medications that can be adjusted. With proper attention to sleep hygiene, treatment of underlying sleep disorders, and management of medical conditions, many older adults can achieve good quality sleep. The changes in sleep across the lifespan reflect changing biological needs. The high proportion of REM sleep in infancy and early childhood may support the rapid brain development occurring during these years. The abundance of slow-wave sleep in children may facilitate growth and the extensive synaptic pruning and organization happening as the brain matures. The reduction in both REM and slow-wave sleep with aging, while partly reflecting declining health, may also reflect reduced need for these processes as the brain becomes more stable and less plastic. Understanding sleep across the lifespan has practical implications. Parents of newborns benefit from knowing that frequent night waking is normal, not a sign that something is wrong. Sleep training methods should account for developmental readiness. Schools might reconsider early start times for teenagers, recognizing that they're fighting against adolescent biology. Adults should prioritize sleep with the same attention given to diet and exercise, and older adults should seek help for sleep problems rather than accepting them as inevitable. Sleep evolves as we age because we evolve, because our brains and bodies have different needs at different life stages. But through all these changes, one constant remains. Sleep is essential, and getting adequate, high-quality sleep at every age supports health, cognition, emotion, and well-being. Closing, the twist. And so we return to the question that opened our journey. Why do we sleep? We've traveled through stages and cycles, through molecules and memories, through the glymphatic rinse and the neural replay of consolidation. We've seen that sleep serves not one function, but many. Consolidating memories, processing emotions, clearing metabolic waste, regulating hormones, supporting immune function, facilitating creativity, and maintaining the delicate balance of brain chemistry. But here, at the end of our exploration, I want to share something that might shift your entire perspective. Something that sleep researchers have begun to whisper in recent years, though it challenges our most basic assumptions about consciousness and rest. What if we've been asking the wrong question all along? For over a century, scientists have framed sleep as something that requires explanation. Why, they ask, would evolution create a state that makes us vulnerable, unconscious, unable to eat, or reproduce, or watch for danger? Sleep seems like a biological puzzle, a mysterious behavior that needs justifying. But consider this alternative interpretation, one that grows more compelling as we understand the complexity of sleep's functions. What if sleep is not the state that needs explaining? What if wakefulness is? Think about what we've discovered. During sleep, your brain consolidates memories, transferring them from temporary to permanent storage, a process that appears impossible during the constant influx of waking information. During sleep, the glymphatic system clears toxic proteins that accumulate during neural activity, operating at twice the efficiency of waking clearance. During sleep, your brain literally expands its extracellular space to flush out waste, a process that requires the unique neurochemistry of sleep. During sleep, emotional experiences are processed and integrated in ways that prevent psychological damage. During sleep, the immune system mounts its strongest defenses and the body performs its most intensive repairs. These aren't minor maintenance tasks squeezed into leftover time. These are fundamental processes without which the brain and body cannot function. Based on what we now understand about the scope and necessity of these processes, we might reframe our understanding. Perhaps sleep is not an interruption of life, but its foundation. Some researchers have begun to suggest a radical reinterpretation of the sleep-wake cycle. In this view, sleep is the default state, the baseline condition of the brain. Wakefulness, by contrast, is the special state, the high-energy, resource-intensive, unsustainable mode we enter to gather food, find mates, and navigate social relationships. We don't sleep to rest from waking. We wake to accomplish the specific tasks that require consciousness, and then return to sleep, the state where the brain performs its essential work. The evidence for this interpretation is circumstantial but intriguing. Developmentally, fetuses spend most of their time in sleep-like states. Newborns sleep 16 to 18 hours per day. Only gradually, as we age and our brains mature, do we spend more time awake. Even in adults, the homeostatic sleep drive, that relentless buildup of adenosine, ensures that we cannot stay awake indefinitely. The pressure to sleep always wins. Every organism that has a nervous system of any complexity shows sleep or sleep-like states, suggesting that once you have a brain, sleep becomes mandatory for its maintenance. From this perspective, the vulnerability of sleep is not a design flaw, but an acceptable cost. The alternative, skipping the maintenance, would be catastrophic. It would be like running a factory at full production 24 hours a day, never stopping for repairs, never clearing the waste, never allowing the machinery to reset. Eventually, the whole system would grind to a halt. Similarly, a brain that never slept would accumulate toxic proteins, lose the ability to form lasting memories, become dysregulated in its emotional responses, and ultimately fail. This reframing doesn't change the specific mechanisms we've explored. The stages remain the same, the molecules function as described, the benefits are as real as ever. But it shifts how we think about the relationship between waking and sleeping. Sleep is not time stolen from life. Sleep is the foundation upon which waking life is built. The hours you spend unconscious are not wasted, they are invested, allowing the hours you spend conscious to be effective, stable, and sustainable. So tonight, as you prepare for sleep, you might consider this new perspective. You're not leaving the world to rest. You're returning to a state of active maintenance and restoration, a state that your brain requires not as a break from its real work, but as the continuation of it. The cleaning, the consolidating, the processing, the regulating. These are not secondary functions happening during downtime. They are primary functions that define what it means to have a functioning brain. Your consciousness, that bright thread of awareness you carry through each day, depends entirely on the unconscious work that happens each night. Memory, emotion, creativity, health, all of it rests on the foundation of sleep. And perhaps most remarkably, this nightly journey into unconsciousness, which we might have seen as a loss of self, is actually how the self is maintained, rebuilt, and prepared to wake again. If you found this exploration thought-provoking, if it changed how you think about those hours in darkness, consider subscribing to the Sleepy Science channel. We bring you deep dives into fascinating scientific topics, each presented in this thorough style, designed to engage your mind as it settles toward rest. Sleep well tonight, knowing that your brain knows what to do. The stages will unfold, the chemicals will balance, the cleaning will proceed, the memories will file themselves away. You don't need to control any of it. Simply allow it to happen. Give yourself the time, create the conditions, and trust the process that has sustained conscious life for hundreds of millions of years. The question isn't why you sleep. The question is, how could you possibly function without it? Rest well. Your brain has essential work to do, work that defines everything you are and everything you'll become. And that work begins the moment you close your eyes. Good night.